for tuning in to Planetterrian Daily, episode 127. It's July 21st, 2026. Some interesting findings to cover today. Phase 1 trial finds iPSC-derived neural progenitors safe in subacute spinal cord injury over 2 to 4 years of follow-up. Ruhr University researchers developed CFREI confocal relaxation imaging to measure folding dynamics of proteins within membraneless organelles in living cells. The method distinguishes whether these compartments promote harmful aggregates linked to Alzheimer's and Parkinson's or temporarily sequester them. Early data show organelles can buffer misfolded proteins under stress before aggregates form. The technique avoids fixation artifacts that previously limited such observations. Most people picture protein aggregates as simple waste that cells should clear immediately. In reality, certain membraneless organelles can temporarily hold misfolded proteins, acting more like holding areas than permanent storage. Right now, inside your cells, these dynamic droplets are constantly forming and dissolving as proteins are made and checked for proper shape. Researchers can now watch folding kinetics in real time within living organelles using a new confocal relaxation imaging method, revealing behavior that fixed samples had hidden. Future therapies targeting protein misfolding diseases may need to preserve rather than always disrupt these organelles, depending on whether they are protecting or promoting aggregation in each context. Now, shifting to a very different area of research. Endothelial cells carrying intrinsic NOD2 drive generation of effector and memory T cells that regulate gut immunity. The work maps how this receptor in vascular cells of the intestine coordinates adaptive responses without relying solely on immune cell expression. Mouse models lacking endothelial NOD2 showed altered T cell populations and weakened mucosal defense. The findings clarify a vascular immune axis that may influence inflammatory bowel conditions. Next steps include testing whether NOD2 variants in endothelium alter disease susceptibility in patient cohorts. On a different note, cells respond to sudden temperature spikes by chemically modifying existing proteins rather than waiting for new synthesis. This post-translational control allows quick on-off switching of protein activity during acute stress. The study demonstrates the mechanism operates faster than transcriptional responses and may protect core cellular functions. Experiments used controlled heat pulses on cultured cells to isolate the modification timeline. Researchers plan to map which protein classes are most frequently toggled by this route. Next up, Oklahoma Museum researchers and collaborators show that standing genetic diversity, not novel mutations, supplies most adaptive capacity when species face rapid environmental change. The analysis examined multiple animal lineages and found pre-existing variants often confer tolerance to new temperature or precipitation regimes. This shifts emphasis from waiting for beneficial mutations toward conserving existing diversity. Field and genomic datasets from threatened populations support the pattern. Conservation strategies may now prioritize maintaining large, genetically diverse groups. A review of existing studies highlights how traditional practices can address climate impacts, biodiversity decline, and nutrition shortfalls simultaneously. Researchers note that integrating local ecological understanding with modern agriculture improves resilience in variable environments. The analysis draws on cases from multiple regions where indigenous methods maintain soil health and crop diversity. No single intervention is proposed. Instead, the work emphasizes context-specific combinations. Future policy work will test scaled implementation in different biomes. QUT team identified molecules that keep carbon nanotubes dispersed, overcoming a 20-year barrier to high-performance thermoelectric materials. Dispersed nanotubes achieved record conversion of waste heat to electricity in flexible formats. The approach targets wearable devices that could power sensors from body heat. Material tests confirm stability under repeated bending. Scale-up trials will assess durability in real-world thermal gradients. City University of Hong Kong researchers demonstrated piezosynthesis, where vibrations split water or generate useful compounds without external electricity. The process uses mechanical energy from waves or flow to activate catalysts. Lab setups produced measurable yields of target chemicals under controlled vibration frequencies. The work opens routes for decentralized fuel or chemical production near water sources. Engineering refinements will test efficiency at larger volumes. University of Texas astronomers propose that early universe little red dots represent precursor structures that evolve into present-day globular clusters. The model links two previously separate puzzles by showing how dense star-forming regions could persist and reorganize over cosmic time. Simulations match observed sizes and stellar densities. Spectroscopic follow-up with upcoming telescopes will test predicted chemical signatures. The hypothesis reframes how the first dense stellar systems formed. University of Illinois researchers established 64 one-acre tile-drained plots to run controlled experiments on soil, water, and crop interactions for the next 150 years. Farmers helped select variables, including rotation systems and input levels. The scale allows measurement of drainage effects on nutrient loss and yield stability. Data will inform regional adaptation to changing precipitation patterns. Annual reports will track both agronomic and environmental outcomes. The Phase 1 spinal cord injury trial using iPSC-derived neural progenitors met its safety goal and provided 2 to 4 years of MRI and functional follow-up data. This adds human evidence to regenerative strategies that aim to replace or support damaged neural tissue after acute injury. It moves the open question of long-term graft behavior forward by showing stable imaging appearance without serious cell-related complications. A Phase 1 trial transplanted iPSC-derived neural progenitor cells into patients with subacute complete spinal cord injury and followed participants 2 to 4 years. Primary safety endpoints were met with no serious adverse events attributed to the cells. MRI and neurological assessments documented stable graft appearance and modest functional signals in some cases. This extends regenerative approaches by providing multi-year human data on cell integration. Longer controlled studies will measure efficacy endpoints. The Genomas Brazil program sequences patients at population scale and integrates results into routine clinical care within the unified public system. Infrastructure includes sequencing centers, data platforms, and clinician training. Early implementation focuses on pharmacogenomics and rare disease diagnosis. The approach aims to deliver precision benefits equitably across a large middle-income country. Evaluation metrics will track diagnostic yield and treatment changes. 10 years of California enrollment data reveal persistent gaps in college preparatory course placement for students classified as English learners. The analysis links classification practices to reduced access even when academic performance would otherwise qualify students. Districts that adjusted placement rules saw increased enrollment in required courses. The findings inform ongoing revisions to multilingual learner policies. Additional states are examining similar classification effects. An International Journal of Business and Systems Research experiment compared ad campaigns and found visual distinctiveness predicted purchase intent more reliably than emotional arousal alone. Participants viewed matched pairs of ads differing mainly in memorability of imagery. Click and conversion data favored the visually striking versions across product categories. The result challenges assumptions that strong emotion is the dominant driver online. Advertisers may now prioritize image testing protocols. City Street George's study of grant recipients found successful female applicants later secured more citations and additional funding than unsuccessful female peers. Male applicants showed smaller differences between funded and unfunded groups on the same metrics. The data cover multiple disciplines and career stages. The pattern suggests early resources compound differently across genders. Longer-term tracking will examine whether these citation gains translate into sustained lab leadership. From regenerative approaches now being tested in human injury, exosomes collected from mesenchymal stem cells reversed aspects of metabolic dysfunction in a mouse model of liver disease. Treated animals showed reduced fat accumulation and improved markers of hepatocyte health compared with controls. The vesicles carry signaling molecules that modulate inflammation and lipid handling without requiring whole cell transplantation. Delivery was systemic and well tolerated in the short term. Work now moves to larger animal models and dose optimization. Keep an eye on larger animal dosing studies for vesicle-mediated signaling in metabolic models. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time, and if you'd rather watch than listen, find us on YouTube at NERA Network. Links in the show notes. One more thing, if you liked today's episode, our sister show Models and Agents for Beginners is worth a spot in your feed. Artificial Intelligence Explained Simply for Beginners and Curious Teens. It's one of the NERA Network's daily briefings, all free at neranetwork.com. Following the long arcs? Story trackers on neranetwork.com follow major programs across episodes, Tesla, SpaceX, AI, space science, and more. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.